Welcome to all. I am Dr. Kadam Asha from Department of Botany, Dada Patil Mahavidyalaya, Karjat. Today we learn about the plant constituent or the cell organelle, that is the ribosome. In the part first, we learn about the morphology of ribosome, types of ribosomes, ultrastructure and the chemical composition of ribosome. So, we know that ribosomes are attached to the endoplasmic reticulum. They are commonly called as the cell's factory because of they play an important role in the site for protein synthesis, and we know that protein is the important molecule. So, ribosomes are called as the cell's factory. They are the site for protein synthesis, that is the process of translation. Means ribosomes play an important role in the protein synthesis through the process of translation. They are also known as translational apparatus. They are the non-membranous organelles. Means when we observe these ribosomes, they having no any membrane bound structure. They are membrane free. Means they are non-membranous organelles. They are only detached to the other organelles like the endoplasmic reticulum. Mostly they are attached to the rough endoplasmic reticulum. Sometimes they are also found freely in the cytoplasm. Here these are the ribosomes which are present freely in the cytoplasm. So these are ribosomes. When we see the diagrammatically, here the ultrastructure of the plant cells in which there are the various organelles are seen. Here these network-like structures that we call as the endoplasmic reticulum. When the ribosomes are attached to these endoplasmic reticulum, that time the surface of these endoplasmic reticulum, they appears very rough, and due to their rough appearance, this endoplasmic reticulum is called as the rough endoplasmic reticulum. And most of the time these ribosomes are attached to these endoplasmic reticulum. Rarely they are freely found in the cytoplasm of the cell. So these ribosomes are very tiny, minute or the dense particles. These are made up of a rRNA, that is the ribosomal RNA, and some proteins, that is the ribosomal proteins. Who discovered these ribosomes? So, first in 1950, the cytologist, that is the George Emil Palade. This is the image of George Emil Palade who discovered ribosomes in 1950. Under the electron microscope, they show that these ribosomes are very small and tiny or the dense particles, and that's why they give the name, that is the microsomal particles. When they observe these dense particles under the electron microscope, that time they observe that these bodies are very small, and that's why they give the name microsomal particles. Micro means minute and doma means body. After in 2000 to 2001, detailed study of ribosomes carried on, and then they are found the different structural component of the ribosomes. On the basis of where these ribosomes are located, there are mainly two types. That is the membrane bound ribosomes and the free ribosomes. These two names itself indicates its meaning. Means those ribosomes which are attached to the some organelles, that is the endoplasmic reticulum or maybe the other organelles, that time we call as the membrane bound ribosomes. But those ribosomes, they are freely available in the cytoplasm, that time that ribosome is called as the free ribosomes. Means on the basis of the location of the ribosomes, they having the two types. That is the first one is the membrane bound ribosomes. Means those ribosomes which are attached to the other organelles. That is the most of the time these ribosomes are attached to the endoplasmic reticulum, and rarely sometimes they attach to the other organelles also. For example, we know that in the systole, flattened structure of the Golgi apparatus, also there are some ribosomes are attached. And second type is the free ribosomes. Means those ribosomes are freely available in the cytoplasm, that type we call as the free ribosomes. So first, when we learn the membrane bound ribosomes, in these ribosomes there is the synthesize. These ribosomes are always synthesize the proteins for the membranes and also the exocytosis. Means they are also used outside the cell. These are always attached to the endoplasmic reticulum and the other organelle. This diagram shows that here there is the endoplasmic reticulum on which there is the attachment of a dense particles that we call as the ribosomes. Here these ribosomes are attached to the rough endoplasmic reticulum, and that's why these ribosomes are called as the membrane bound ribosomes. Then next is the free ribosomes. Means these ribosomes are freely available in the cytoplasm. Means in the cytoplasm of the cell, when these ribosomes are freely available, that time we called as the free ribosomes. These free ribosomes are synthesize the proteins which functions always inside the cytosol. Means they are used inside the cell. For example, metabolism of some food particles. So these are the two types of ribosomes, that is the membrane bound ribosome and second one is the free ribosomes. Membrane bound ribosomes means those ribosomes which are attached to the rough endoplasmic reticulum or the another organelle, and free ribosomes means which are available freely in the cytoplasm. Both these ribosomes are synthesize the proteins, but membrane bound ribosomes, they synthesize the proteins for the membrane and the exocytosis. Means they are used outside the cell. Here free ribosomes, they synthesize the proteins but they function inside the cytosol. Means they used inside the cell, for example metabolism of a food particles. When we learn the structure of ribosomes, these ribosomes are made up of the rRNA, that is the ribosomal RNA, and some distinct proteins, and that distinct proteins we call as the ribosomal proteins. So there are approximately in the prokaryotes there are the 3 rRNA, that is the ribosomal RNA are the 3 in the prokaryote, while as in the eukaryote there are the 4 ribosomal RNA and 40 to 80 different proteins. Means when the ribosomes they are made in the prokaryotic cell, that time there are 3 rRNAs and the 40 to 80 different proteins, and when these ribosomes are made made in the eukaryotic cell, that time that made up of a 4 rRNAs and 40 to 80 different proteins. So these structure or these ribosome, they are arranged in two pieces. These image, when we learn this image or the picture of the ribosomes, that time here we see that there are two pieces of the ribosomes. So they are arranged into two pieces. While translation means when the translation process is carried on, that time these ribosomes are arranged into the two different pieces. That is the small ribosomal subunit and the large ribosomal subunit. Sometimes it also called as the smaller ribosomal subunit, and these are the larger ribosomal subunit. When we learn the larger subunit of ribosome, that time it contains the active site of ribosomes. Means large subunit of ribosomes that always contains the active site of ribosomes. The site where the new peptide bonds are formed. Means these diagrams showing the larger subunit of ribosomes, which is the active site of ribosomes. The site where the new peptide bonds are formed, these showing in the green color. This green color show that here is the site where the new peptide bonds are formed. Then these large subunit of the ribosomes also contains amino acid and the peptide binding sites. It uses the peptide transferase enzymes to catalyze the process. Here this green color that showing the sites for where the new peptide bonds are formed. So here there is the enzyme, that is the peptide transferase, which play an important role to catalyze the process of the translation. So there are made up of two rRNA strands in the larger subunit of ribosomes. There are the two strands of ribosomal RNA, that is the rRNA, that is here in the diagram showing the yellow and the orange strands. These yellow and these orange strands, they made up of these rRNA strands and also the protein strands. These blue color showing these are the protein strands, means these yellow strands and orange strands, they are the rRNA strands, while as blue strands in this diagram, blue strands indicate the protein, while as yellow and orange strands indicate the rRNA. Means large subunit of ribosomes is made up of these rRNA and the protein. So here this diagram showing the structure of the ribosomes, in which the protein synthesis is carried on. So here there are the different sites. Peptide binding site, these are the exit site, and these are the transfer RNA molecules. When there is the formation of a polypeptide chain, that these showing the growing polypeptide chain. We know that ribosomes having two subunits, that is this one is the smaller subunit and this one is the larger subunit. So here these are the amino acid binding sites. Here these are some messenger RNA terminals, nucleotide units, and here in these process Means in this is the translation process in which ribosome play an important role in the synthesis of a protein. So, when we learn about the small subunit of the ribosomes, we say that it also play an important role in the process of a translation. So, in charge of a information flow, pairing the codon with the anticodon, and in it always intake the messenger RNA, that is the mRNA. This messenger RNA then enters through the opening in the left side and it's made the ribosomal RNA. This orange strand showing the ribosomal RNA and this blue strand showing the protein strands. So, it contains the entry and exit of the messenger RNA. Means these small subunit of ribosomal RNA, they also contains the strand, that is the protein strand, and these are the rRNA strand. Means smaller subunit also made up of a protein and the rRNA strand, which play an important role in the entry and exit site of a mRNA, that is the messenger RNA. So, these are some references regarding the ribosomes, in which we study the introduction of ribosomes, then different types of ribosomes on the basis of their location, and after we learn the structure of ribosomes, in which ribosomes having the two subunits, that is the smaller and the larger subunit. We know that in the smaller and the larger subunits, these are made up of a two strands, that is the protein strand and the rRNA strand. Thank you.